Hello, Professor. Today we're presenting the parvoviruses and aphids, the virus host symbiosis, typically required for wing development. Today presenting is James Green, Emilio Gutierrez, Sahari Mendoza, and Serafina.
Parvoviruses is a family of small, rugged, genetically compact DNA viruses, also collectively known as parvoviruses. They are linear, non-segmented, and single-stranded DNA. Some important characteristics that come with these viruses are that, one, they can infect and may cause diseases in many animals, from arthropods such as insects and shrimp to starfish. Also included are mammals, which also involve us humans. Because most of these viruses require actively dividing cells to replicate, the type of tissue infected varies with the age of the animal. The structure of the virus is very different depending on what the host is, which I will get in more depth later. Parvoviruses are highly pathogenic and kill approximately 90% of the host larvae within days, making them potentially effective as selective pesticides. This may possibly be a result of their extreme reliance on host function, which have prompted their use as highly specific pest control agents. Like I said in the previous slide, the host varies. P. aphids exhibit a textbook example of phenotypic plasticity. Phenotypic plasticity is the ability of one genotype to produce more than one phenotype when exposed to different environments. Therefore, crowded conditions trigger the production of winged rather than wingless offspring. Organisms often respond to changing environments by altering development of particular traits. We identified two laterally transferable genes that contribute to variation in a classic example of phenotypic plasticity. The aphid's ability to produce winged offspring in response to crowding. Our results uncover a novel role for co-opted viral genes, demonstrating that they are used to modulate ecologically relevant plastic phenotypes.
What is the beneficial effect of the virus for the host? Wing morphs are essential for the survival and dispersal of aphids. When comparing the two in an experiment, the aphids with wings were able to outcompete the ones without. They were able to colonize new plants, therefore have access to new resources. The virus increases the chance of survival by promoting wing development. When comparing the two, the ones with the virus were able to grow wings. This is a textbook example of natural selection, adaptations of wings to maximize fitness. In figure 1, it shows the increase and dispersal of virus-infected aphids A, B, and C. A being the original plant, to C being new colonizing plants.
What is known about the mechanisms of parvoviruses and aphids that determine wing morphology? We know that the upregulation of two specific genes is the responsible mechanism for creating winged aphid offspring. These two genes are called APNS1 and APNS2. The presence of this viral domain in winged offspring suggests that the genes are upregulated as a result of lateral gene transfer. These non-structural proteins of parvoviruses will activate transcription factors, which cause these epigenetic modifications through histone acetylation. In the figure to the right, you will see that winged offspring have a much stronger upregulation of viral genes as opposed to wingless offspring.
The interaction that it goes through. This is a mutualistic symbiotic relationship between the parvovirus and the aphid. The virus benefit. The aphid is providing a place for the virus to survive and proliferate in. Without host machinery, the virus would not be able to replicate. The benefit of it. Aphid genes of viral origin or upregulated in response to crowding, causing the aphid to produce winged offspring. As we may also know, parvovirus is very species specific. There are at least 3 well-known types of parvovirus, and the different types of parvovirus infect different species aphids in similar ways. It is believed that this first interaction came from rosy apple aphids.
Thank you all for sticking with us, and that concludes our presentation on host virome interactions between parvoviruses and aphids.